Well, welcome back. This is Michaela and this is Robbie, and they're going to present the soil respiration in terms of carbon efflux.
All right, so for the last 5 weeks now, we've been working with autotrophic and heterotrophic respiration, and I'm Robbie.
And I'm Michaela. So the basis on our research was we wanted to look at the different carbon fluxes trees put out, and we also wanted to see if bacteria and fungi had an effect on it. We also wanted to see if photosynthesis was affected by bacteria, so whether or not a tree could live in sterile soil. So we analyzed both of these by using the LICOR.
And then another thing we wanted to look at was the soil microbes. We wanted to see what kinds of bacteria and fungi were contributing to soil respiration and how much they were contributing. And so basically we took samples of soil and we diluted them down, and then we put them in agar plates and let the bacteria and fungi grow, and then we counted them.
So for respiration is a really big part of ecosystems. Trees put off 60% of respiration. And we also learned that bacteria and fungi do have an effect on soil, so it's not just the trees. And you'll have different fluxes throughout winter and summer because of the dormant trees and the non-dormant trees. You also have the two categories of microbes in the soil are autotrophic and heterotrophic. Heterotrophic put off more carbon during the growth season.
So why this is important? So the carbon cycle has a very big impact on climate change, and so it's important to know how it affects ecosystems and how these processes affect the carbon cycle itself. And we also wanted to see how plants could react to fires that would run through ecosystems, because as that happens, it takes out a lot of the soil microbes, and we wanted to see if the photosynthesis was affected by that.
So our methods, we had 14 different tubes. We had 3 no bacteria, no tree, 3 bacteria with a tree, 3 with sterile soil and a tree, 3 with sterile soil and without a tree, and 2 tubes of vermiculite. So we put the non-sterile soil in the tube, and then we put the trees in. But with the sterile soil, we had to autoclave it first, which is kind of like a big pressure cooker. So it brings down the pressure and it brings up the heat, so making sure that everything is dead by the time it's finished. Once we had that done, we rinsed the PVC pipes with 70% ethanol, making sure those were also sterile before we put the soil in. Then, the ones with trees, we dipped the trees in 70% ethanol because we want those to be sterile as well, but we don't want to kill them. So you couldn't autoclave a tree because it'd be killed. Once that was done, we let them sit for a few days so that their carbon fluxes got back to normal.
Okay, and so this is basically how we set up each tube. They were about 2 inches in diameter of PVC, or polyvinyl chloride. And so we'd first rinse it in ethanol to make sure it was sterile, and then we'd fill it up with soil, and then the tree would just hang out the side there. So we set it up where the blue tape was the sterile soil and the orange tape was the non-sterile soil. And so collecting the data, we used a machine called the LICOR, and so that's it right there. So it has a chamber on the side that would collect the carbon that was given off by the soil microbes. That's called the soil chamber. So it would collect the carbon and send it into an infrared chamber, and that would analyze how much gas was in there, specifically carbon. Then it would send it over here, and we would be able to analyze it. And The LiCor would also be able to measure photosynthesis. You would just have to switch off the soil chamber and put in an LED light chamber, and you would be able to measure the photosynthesis. And so, collecting the data, we just set it on the PVC tubes, and then it would start collecting carbon.
Once that was done, we started taking our samples so that we could grow our own bacteria. So we took a vessel that holds 450 milliliters of water, and then we took 5 grams of soil from each tube, and then we would vortex the vessel for 4 minutes. And by doing this, we just wanted to make sure everything was mixed up so you didn't have your soil sitting on the bottom and the water on top. Once we did that, we took 1 milliliter of water and put it into a test tube with 9 milliliters of water. We went down like the picture shows to 10 to the minus 8 so that by the time you got down there, it was a low enough microbe count that you could actually count it. Because if you started with the vessel that held 450 milliliters, there's no way you could count it.
And these pictures are showing basically the process we separated them to the 4 different treatments, and then we started at 10 to the minus second, diluted it all the way down to 10 to the minus 8, and then once we went through the dilution, we would use the tubes that were concentrated at 10 to the minus 6, 10 to the minus 7, and 10 to the minus 8, and we would put those in the agar plates. And so we incubated those plates for a couple days, and that's how the bacteria and the fungi grew, and then we were able to count them.
And we also put the fungi on a nutrients agar so that it would help it grow, and we put the bacteria on a ZZA agar so that that would grow. Once we left our bacteria a few days, we started to count it. So each dot you see on the Petri dishes isn't one microbe, it's called colonies, so there's thousands of them. So once we counted every dot on the Petri dishes, we put it into a graph. We put everything we analyzed from the bacteria to the carbon fluxes to the photosynthesis into graphs.
So for our results, we saw that there really was no effect that the bacteria and fungi had on photosynthesis. So basically, in each treatment, the level of photosynthesis was basically the same. Then we saw a little bit of a flux within each treatment for soil respiration, but not really noticeable. The most was in the tubes without trees and bacteria. But when we did a bacteria count of each of the tubes, we actually found that the sterile tubes, which we autoclaved at the beginning, they actually had the most bacteria within them. And we think this is because the tubes sat in the greenhouse for so long with other plants that were non-sterile. We think the bacteria could have gotten in through that way. And so we saw that there was a little effect on soil respiration from the microbes.
So for our conclusion, we found that fires don't have an effect on the photosynthesis of a tree. So if you have a fire go through an ecosystem, it will be able to replenish itself and come back, which if the trees couldn't have lived in the sterile soil, they would not have been able to come back. We also learned that bacteria and fungi do have an effect on the carbon output, which is interesting because most people think it's just the trees. And we also learned that the different effects of the photosynthesis and stuff. This is our microdensity chart. So the A's on all the charts signify that they're statistically equal. They look different, but they're not. It showed us that the density of the soil in every tube was the same.
Okay, so this, again, is showing the photosynthesis of each of the treatments. It looks pretty different, but again, they're statistically similar. And if you line up the averages of each of the bars, they basically line up. So again, that means that if a fire was to wipe out most of the bacteria and fungi of an ecosystem, the plants would still be able to thrive or at least function. And so this is showing the the soil respiration of each of the different tubes. And so most of them are statistically similar. We did see one outlier, and that was the tubes without bacteria and without the trees. And so, again, that was the one that had the most bacteria. And so we think that the result came out so high because we measured it at the highest population of the bacteria. So that would lead to the highest flux in the soil respiration. So these are just our references. So we'd like to thank Excel Energy for sponsoring our research. We couldn't have done this whole process without them.
We'd also like to thank Lori Ball for putting this together and helping us figure out everything that a college gives us. We also want to thank Eric and Kai and Scott for taking their summer to teach classes and teach us a little bit about what they went through during college and the different things they learned.
We'd like to also thank our mentor, Mr. Dan Beverly. He helped us throughout this whole process. He helped set up the experiment all the way through the PowerPoint, and so we couldn't have done this without him either.
It was definitely a great learning experience that only he could have given to us, so it was really beneficial. Does anybody have questions?